Hello everyone. Today in the physiology of the endocrine system, we discuss about the general principle and the mechanism of the endocrine hormones. Now first of all, what are the endocrine hormones? They are the secretory product of the ductless glands which are released in the catalytic amount in the human being body in the bloodstream and transported to the specific target cell organs where they elicitate or particularly function physiological, morphological, biochemical and do their response. Now the classification of the endocrine hormones. Now it is basically classified into chemical nature, on the basis of chemical nature and the mechanism of their particular actions. Now first of all, on the basis of the chemical nature, they are divided into mainly three subtypes or subclassified into mainly three types. That is first of all the amine or the amino acid derivatives. In that we have catecholamines and T3 and T4. Now next is the protein or the polypeptide containing hormone, that is the posterior pituitary hormone. In this we have particular two hormones, that is the ADH or the antidiuretic hormone and the oxytocin. Then the insulin, glucagon, parathormone and the anterior pituitary hormones. Now to remember all the hormones of the anterior pituitary gland, we have the mnemonics flat peg, that is F means FSH, L means LH, A means ACTH, T means TSH, for P prolactin, E for endorphins and the G means the growth hormones. Now the steroid containing hormones are the steroidal hormones are the glucocorticoids, mineral corticoids, sex corticoids, vitamin D and the aldosterone. Now classification on the basis of the mechanism of their action. We have mainly two groups, that is the group 1 and the group 2. Now in the group 1 we have basically such hormones that acts by binding to the intracellular receptors, that is the steroid, tretinoid and the thyroid. In the group 2 we have such type of hormone that actually act by that they have particularly secondary messenger with them and they may depends on them for a particular mechanism and for the particular action. For example, in this we have further subgroups in this. For example, group 2 has basically such hormones which are dependent on the secondary messenger for the particular action. And in this we have these subtypes, that is the group 2A, group 2B, group 2C and group 2D. Now in the group 2A we have secondary messenger as cyclic AMP. Now over here the hormones are the ACTH, ADH, angiotensin 2, calcitonin, CRH, catecholamines. In that we have alpha 2 adrenergic, FSH, glucagon, LH, PTH, somatostatin and the TSH. Now in the group 2B we have the secondary messenger as cyclic GMP, and now over here we have the hormones are the atrial natriuretic factor and the nitric oxide. In the group 2C we have the secondary messenger as basically calcium or the phosphoridyl inositol or both. Okay, remember we have over here as the calcium or the phosphoridyl inositol or calcium and phosphoridyl inositol both. Now over here the hormones are the ACH or the acetylcholine. Catecholamines over here, remember alpha 1 adrenergic, gastrin, oxytocin, then the TRH, then the GnRH, PDGF and other substances. Now in the group 2D, over here remember the secondary messengers are the kinase or the phosphatase cascade. Remember, kinase or the phosphatase cascade. Over here the hormones are the human chorionic somatotropin, that means the HCS, erythropoietin, growth hormone, insulin and the insulin like growth factor, that means the IGF-1 and the IGF-2, nerve growth factor, prolactin, etc. So two groups over here, group 1 that actually bind with intracellular receptors and the group 2 have the secondary messengers. Now the very important topic and frequently asked question in the exam, that is the mechanism of the action of the hormones. Okay, now we have basically four mechanisms for the particular action of the hormone, that is the action through the change in membrane permeability, then the action through the effect on the gene expression by the binding of the hormone with the intracellular receptor, means group 1. Okay, just remember in the second type, in the second mechanism we will be having the group 1, because over here as you can see intracellular, and in this we studied that in the group 1 they bind with the intracellular receptors. Now in the third part we have action through the secondary messengers. Of course in the group 2 we have the secondary messengers. So through the secondary messenger which activate the intracellular enzymes when hormones combine with the membrane receptors. And the last is the action through the tyrosine kinase activation. Now the first is the action through the change in the membrane permeability. Now over here certain hormones bind with the receptor present in the cell membrane and cause a conformational change in the protein of the receptor, resulting in either opening or the closing of the ion channels, that means sodium, potassium or calcium ion channels. And as because of the opening of these channels, this ion actually moves and causes a subsequent effect. For example, adrenaline, non-adrenaline, etc. Now next is the action to the effect on the gene expression by the binding of hormone with intracellular receptors. Over here, remember, group 1 hormones will be in function. Now group 1 hormone act by the effect on the gene expression, which include the steroidal hormones, retinoids and the thyroid hormones. If you remember, we discussed about this. Now these hormones are lipophilic, means they actually attract towards the lipid, means they are actually permeable towards the lipid in nature and can easily pass across the cell membrane. Now they act to the intracellular receptor located either in the cytosol, or in the nucleus. Now the events over here, total 7 events occurs. Okay, we'll discuss about individual. Now first of all, first is the transportation. After secretion, the hormone is carried to the target tissue on serum binding protein. This is the serum binding protein and this is the hormone. So transportation occurs. Now in the second part, in the second over here, as you can see, there is internalization. Okay, being lipophilic, I told you before, only they are lipophilic, so the hormone easily diffuses into the plasma membrane. Now in the third part, it is the receptor hormone complex. This is the receptor and this is the hormone, which actually came in the internalization process. So what happens is, there is a complex formation and they mainly form by binding of the hormone to a specific receptor inside the cell. Now fourth is the conformational change occurring. Now what happens over here is, it mainly occurs in the receptor proteins, leading to the activation of the receptor. So because of this, the receptor actually activates. Now in the fifth, as you can see, there is the activated receptor hormone complex that diffuse into the nucleus and binds on the specific region of the DNA known as the HRE. This will bind to the HRE of the particular DNA. The HRE is the hormone responsive elements which initiate the gene transcription. Okay, transcription is basically for production of the mRNA. Okay, it will initiate the mRNA production or transcription. Now the sixth is the binding of the receptor hormone complex to the DNA alters the rate of transcription of the messenger RNA. And over here, as you can see, the seventh, the mRNA diffuses in the cytoplasm. Okay, over here, after coming out, actually it will come out into cytoplasm from the nucleus. Okay, where it promotes the translation process at the ribosome. Translation basically for the production of the protein. Okay, so they actually promote the translation process at the ribosomes. Now in this way, new proteins are formed which result in the specific responses. Some of the new proteins synthesized are the enzyme protein only. Okay. Now next are the action to the secondary messengers. Of course, the group 2 hormones which activate the intracellular enzymes when hormones combine with the membrane receptors. Now over here, the release of secondary messengers by the group 2 hormones occurs, right? And this is actually mediated by the GTP binding protein, which is known as the G protein. Over here, G protein is very important. Now coupling by the G protein is very important. Now what is coupling of the G protein? By coupling by the G protein. Now over here, the events involved in coupling by G protein which lead to changes in the cellular concentration of the secondary messenger are, first of all, the group 2 hormones. The group 2 hormones are water soluble and bind to the plasma membrane of the target cell via the cell surface receptor. This is receptor and they will bind to the membrane by this receptors. Now the hormone bearing receptor then interacts with the G protein. Over here, it actually interacts with the G protein and activates the inactive G protein into active G protein. And it basically activates it by binding the GTP. Now there are basically two classes of the G protein, remember, that is the GS and GI. What is GS? GS is the stimulatory G protein and the GI is the inhibitory G protein. Now in the activated or the on state of the G protein, the G protein interacts with one or more of the effector proteins. It will actually interact with these inactive effector molecules or the proteins, most of which are enzymes or ion channels such as the adenyl cyclase, calcium or the potassium channels, or phospholipase CA2 or D. So now what it will do with the interaction of this particular inactive effector molecule, after interacting with these, it will actually activate this effector molecule and forms the active effector molecule. Now the changed effector molecule in turn generates, remember, it will generate the second messenger that mediates the hormonal intracellular action. So this basically coupling by the G protein is basically done to increase the amount of, or we can say increase or generate the secondary messengers, and this secondary messenger will actually cause the activation of the intracellular enzyme when the hormone combined with the membrane receptors. Okay, so this was the main production of the secondary messengers. So now just remember, when the hormone actually combined with the membrane receptor, or the receptor which is present on the outer surface of the cell or on the cell membrane, okay, when they actually reach or combine with them, what will the secondary messenger will do? They will actually activate the intracellular enzymes and will help in the interaction and the particular mechanism of the action by the hormones. Now, which are the secondary messengers in the bodies are the, first of all, CAMP, adenyl cyclase, CAMP system, guanyl cyclase, GMP system, calcium carbonylation system, and others are the membrane phospholipase phospholipid system. Now next is the activation through tyrosine kinase activation. Now over here the group 2D hormone act by tyrosine kinase activation. What were the group 2D hormone? There were the HCS, then the erythropoietin, growth hormone, insulin, IGF, prolactin, etc. They actually act by tyrosine kinase mechanism. Now over here remember the secondary messenger are the kinase or the phosphatase. The activation of the tyrosine kinase occurs by the following two mechanisms, that is the hormone receptor possessing the intrinsic tyrosine activity and the hormone receptor that do not possess the intrinsic tyrosine activity. Now for this, look at this diagram. As you can see, this is for the hormone receptor possessing the intrinsic tyrosine activity, and this is for the hormone receptor that do not possess the intrinsic tyrosine activity. Now over here the examples are the insulin and the epidermal growth factors. Now what happens over here is the binding of the hormone to the receptor changes its conformation and exposes the site on its intracellular portion that are capable of receptor autophosphorylation at specific tyrosine sites. Now as a result of this, what happens is the receptor itself becomes a tyrosine kinase, okay, that phosphorylates the tyrosine residue on the intracellular protein substrate. Now this later activity sets into the motion as a cascade of events leading to the enzyme activation and the gene expression. Now next is the hormone receptors. They do not possess the intrinsic tyrosine activity. Now over here the example are the growth hormone, protein releasing hormone, and the cytokines. Now over here the hormone actually bind with the receptor surface, and because of the binding they actually causes the conformational change in the receptor, that is the intracellular plasmic tail of the particular receptor actually changes. Now because of this change actually causes the attraction and the docking of the intracytoplasmic tyrosine kinase. Okay, now this is actually done with the help of JAK2. What is JAK2? JAK2 is the Janus tyrosine kinases. Now because of this there is the docking and the attraction of this tyrosine kinase. Now because of this there is conversion of the inactive tyrosine kinase to the active form, and after this there is the phosphorylation or transcription factor proteins, and this is basically converted into modulated gene expression by the help of the STATs. What is STATs? STATs is the signal transducer and activator of the transcription kinase, and because of the modulation of the gene expression there is actually action of the hormone occur inside the cell. So this was all about the mechanism and the general physiological structure and the features of the endocrine system. We will discuss about all the types of endocrine hormones and the endocrine glands in the further next videos. So hope you understood well. Thank you so much.